So I use SynWiki all the time. It's very useful for me with my work. So I am Adam Belsom, and I'm working in the group of Yuri Rapsilber. Here in Berlin, we are using and developing a technology called cross-linking mass spectrometry. So what is cross-linking? So essentially, cross-linking is a means to translate proximity into covalent bonds, which we can then read out with mass spectrometry. So we're answering the basic question of which residues are near each other. So there are two aspects to our workflow. So there's an experimental aspect and there's a computational aspect. So first step is you need to choose a cross-linker, or you need to choose the chemistry that you want to use to do the cross-linking. So typically we use these chemical reagents called cross-linkers, and they are consisting of a spacer separated by reactive groups at each end. And these reactive groups can vary, but they react with different amino acids. And the spacer can be, so the spacer acts as a distance restraint, but the spacer can be inert, as in this case, which is a simple alkyl chain, or you can have added functionality added onto the spacer that will help your cross-linking MS analysis in some way. So first of all, you take the cross-linker and you put it on your proteins of interest or purified complex. And this is very, very easy to do. So basically any lab anywhere in the world can do this. And then we then digest the proteins using enzymes, at which point we've lost all the three-dimensional structural information of the protein. However, that information is maintained in our cross-links. So we have cross-linked peptide pairs. So we have this very complex mixture of cross-linked peptide pairs, and also we have linear peptides, which we then spray into the mass spectrometer, and we get thousands of spectra. So then comes the computation. So we use some quite heavy database search to match our spectra from the mass spectrometer to the spectra predicted by the sequences of proteins in our database. Then when we've done that, it's very important that we apply an FDR filter, so a false discovery rate procedure, so that we can be confident with the data that we're producing. And we can estimate what is the error in our data. And then the next step is the visualization. And we then use the data for model building and testing. So that's the workflow. And it works extremely well for individual proteins and also for complexes. But we are now pushing this technology into even further. So we're pushing it into lysates, organelles, and intact cells. And for this, we want to identify complexes, and we want to identify protein-protein interactions, and also identify the interaction interfaces. So this presents new challenges. So large-scale cross-linking presents new challenges. One of the challenges is actually getting cross-linkers to enter the cell. And then because of the complexity that you have, and also the low abundance of cross-linked peptides that you're interested in, you then need to employ some fractionation to simplify your analyte. Then obviously the database search also becomes more difficult because when you increase the number of proteins, and then you have the visualization, which also, how do you display like these networks? But luckily, we have done some nice development for the visualization aspect. So our lab in Scotland has worked on this software. So it's a web-based software for visualizing cross-linking MS data. And actually, so I can also link with SynWiki. So I can export the data from SynWiki and I can upload it into CView. And so we can get extra information on the proteins that we're looking at. We can also upload PDB files so we can fit where the structures are available, so that we can fit our cross-linking data to the structures. So we had some success with in-cell cross-linking. So this was published last year in Science in collaboration with our friends at the EMBL, so Julia Mohammed and Liang, and our friends at Göttingen, so Jörg and Neil. In this study, we produced a network, and in this network we saw interaction. So it was an unexpected indirect interaction between RNA polymerase and ribosome via NUS-A. And then combining the cross-linking data with cryo-EM, we were able to match the density from the cryo-EM to NUS-A. So it could really be shown that there was this coupling of transcription and translation in Mycoplasma pneumoniae. So then through our collaboration with Jörg and Neil, we also had the opportunity to work with Syn3A. So we were very excited about this because it's a minimal genome, and also for us, for the database search aspect, this means that there are fewer proteins that we need to worry about. So for this, actually, we have applied our latest development, which is now we have synthesized and developed a new cross-linking reagent, and this cross-linking reagent has an enrichment, an affinity group, which allows you to enrich cross-linked peptide pairs, and it's cell permeable, so it goes inside the cell, and we get cross-linking throughout the cell. And what does enrichment mean? Well, one of the problems that we have with our method is that cross-linked peptides that we're interested in, they're very, very, very low abundant. And also we analyze this as a very, very complex mixture of peptides, and within this mixture we have the linear peptides, which are the most abundant analyte, and which we're not actually interested in for our purposes. So in our total sample, what we match in the mass spectrometer is only 1.7% of what we match is actually cross-linker modified. However, the impact of enrichment is that after enrichment, we have now 99.6% of the peptides that we identify are cross-linker modified. And furthermore, we can increase the intensity of what we match by 1,000 times. So this means that we can go much, much deeper with our analysis. So we applied this cross-linker and our new workflow to Syn3A, and this is the preliminary data that we have. So we have this small network. We have over 6,000 heteromeric links. So these are links between different proteins. And we have just under 15,000 self-links. So these are links that are within proteins. And we have cytosolic proteins cross-linked together, and we have membrane proteins shown here in blue. And also what is nice is that within the membrane proteins, there are several unknown and uncharacterized proteins in our network. And I think Neil is going to talk a bit more about this in the talk that's coming up. But overall, we identified 426 proteins in Syn3A, and we identified 386 with cross-links. And we identified cross-links in known complexes. So we have the ATP synthase, and we have lots of cross-links in the ribosome, which I'm going to talk about in a second. But I've grouped the subunits of ribosome here. So those are clustered together. So focusing on these unknown proteins, we have this network here. So they're very clearly interacting, and also we don't just know that these proteins are interacting somehow, but we also have the residue level information for this. And then furthermore, we also have the links within the proteins. So we have also data that we can start to use for modeling of these proteins. So this is something that we are now looking at, and it's ongoing work. And also, where we have structures, we can fit them to the known structures. So here we just have cross-links in FTSY, which is a relatively small protein, and we have cross-links that show cross-linking between the subunits of ATP synthase. So this is what we have so far. So far so good. However, when we looked at the ribosome crosslinks, it looks quite messy. So here I'm just showing the crosslinks that are between the subunits of the ribosome, but also the crosslinks that are within the subunits, they also look quite messy. So if we look at this this histogram on the right, so we have the distance distribution. So we're looking at the C alpha distances, C alpha to C alpha distances between our cross-link residue pairs. And in the gray is the distribution that we would expect if cross-linking were random. However, the links that we get for our heteromeric links, they fit this this distribution. So it suggests that this is not what we would expect if everything is working nicely. And when we look at the data that we got for Mycoplasma pneumoniae, we got a completely different picture. So in this case, the links between subunits, they fitted quite nicely. So this is something that is puzzling us at the moment, and it's something that we would be interested in finding out what could be happening here. Is this noise in the cell, or basically what is happening here? This would be very interesting to know, and that's where I will finish. And so this is my group in Berlin, and thank you for listening. And any questions, please?
Our proteomics data, Adam, shows that where I know this is the case in other cells, but that or at least what we were able to detect using proteomics of the ribosomal proteins is, you would think that they would all sort of be expressed at nearly the same level, but that's certainly not the case. And we could have messed this up some by removing, in our efforts to remove genes that may have screwed up transcription or translation, to make SYN3A levels of ribosomal proteins different than you would have found in the wild type Mycoplasma mycoides. So you could have ribosomal proteins that have formed transient because they're not present at the normal levels relative to one another. That might be a reason that you're seeing some of these differences, but I'm really curious how this is going to pan out, because I was hoping you were going to say that your interactome matched brilliantly with the predicted ribosomal structures.
Yes. Yeah. So now as a control, we're actually we've cross-linked SYN1. So we're curious to see what this looks like, because the data for SYN3A, it was a surprise because for Mycoplasma pneumoniae, so we have fewer cross-links. However, they fit very nicely. So yeah, this was a big surprise. So yeah, so we're going back and we're going to look to see what SYN1 looks like, to see if it's a result of the minimized genome. But yeah.
Where possible, how often does the pneumonia interactome agree with the SYN3A interactome?
So I haven't looked at that. I haven't looked at that in huge detail. I mean, we have fewer. I mean, with this analysis, it's a much deeper analysis. So it's possible that we are capturing. I mean, at the moment I mean, it's 3 times more heteromeric links than we got with Mycoplasma. And it's possible that we have we could be capturing noise in the cell just by going deeper, but although I don't think that's the case, because we don't see, for example, the ATP synthase, we don't see all the subunits cross-linked together. So, I mean, I think there is overlap, but we also see some differences too. Yeah, but this is something that I will look at in the future. So,
in the penultimate talk today, Elizabeth Villa will be showing cryotomograms of SYN3A. And she has made the comment to us that the SYN3A cytoplasm seems denser than, more protein packed than with other bacteria that they have looked at using the same techniques.
yes. So we see these hub proteins in the cytosol, and these seem to be cross-linking to everything. So here we have TIF-A. TIF-A is cross-linking, yeah, basically everywhere. So, and this was also something we were thinking about the ribosome. So is this an issue with with density? But and then because Mycoplasma pneumoniae is much slower growing than SYN3, so is it a question of SYN3 producing the ribosomal proteins as quantity rather than quality? But yes, it would be very interesting to see the electron tomography. Yeah.
Okay, well, now for Interactome Part 2. Neil, you're up.
Okay.
Can you see my screen?
You're good. Put it
Yeah.
in projection
Alright, cool.
mode, please.
All right.
Great.
So
Super.
I will carry on now with protein interactions, and I'm going to branch off from where Adam left off, to be honest. And I work in the lab of Professor Jörg Stulke here in Göttingen with Yuri and Adam and Henning and Alexander from Göttingen as well. And just briefly, we are interested, why we got into this project is because we were interested in the unknown proteins that were found in this synthetic cell as well as the generic, so the known unknown proteins. And this team here in Göttingen was initially concerned with Mycoplasma pneumoniae and its function as a minimal cell and as a pathogen. And this Mycoplasma group is seeded in Bacillus subtilis group. So we have a lot of techniques that are common and a lot of history or experience characterizing unknown proteins. That's what I was doing in Mycoplasma pneumoniae previously, and in the publication in the study that Adam mentioned. And then we had the opportunity of syncing up with SYN3, and this was awesome because it grows faster and there's a more vibrant community. And this is the same picture that's behind John on the wall. And if all the genes that are present in this genome are essential or quasi-essential, then deleting them and generating phenotypic mutants becomes a problem. Therefore, we turned to protein-protein interaction maps. This is an older map from what Adam showed you. So you can already see how things improve and how the clarity or the resolution of these maps increase over time. And it's really been a wonderful process working with Adam and Anna to develop these interaction maps and crossing the cells and tweak things as we go along. But what I want to do is draw your attention to this subset of proteins, which is these three proteins 439, 440, and 505. They are all three unknown proteins. And we found that they are heavily cross-linked right from the first few maps that we started seeing, both internally and within each other. And we wanted to start characterizing this complex. At that point, we didn't know what we were dealing with. Turned out they were membrane proteins. And it turned out that 440 was predicted in this paper here from the Zhang lab as a binding protein that's somehow involved with cell adhesion maybe. And we set about trying to express these proteins to clone and codon optimize them before we knew of the existence of the Free Genes project. And coincidentally, two of these genes are not in that project, in those 398 genes. So it's good that we still persevered with them, but it was a lot of work because to optimize them for expression in E. coli is challenging. And I changed this slide a bit because we were trying to figure out what this protein does, and we knew from Mycoplasma pneumoniae that you get certain vesicles outside the cell membrane. So this is a Mycoplasma pneumoniae cell attached to a surface. These are ribosomes you see here. This is the lipid bilayer, and you have these little dots that are always present on the cell surface in this cryo-electron tomograph image. And we had the hypothesis that these set of proteins, because they are membrane localized and they are unknown in function, had something to do with anchoring these vesicles here. And we are working towards that and trying to figure out what they're doing. And we also could take the crosslinks that were predicted from our maps and map them onto predicted structures for 439 from the Zhang paper and from other protein structural prediction tools. And some of them match quite well, as you can see in green, and some in red don't match at all. But this is a very large protein and has several flexible regions, so as to speak. But I would take a minute out and change my talk slightly, because based on what Andre and Chris already mentioned, it seems that these proteins could also have a role in RNA binding and therefore in the problems that they've had in their cell-free setup systems. We know this because we also tried to make RNA protein interaction maps here, because there's a bunch of publications that deal with how metabolic enzymes moonlight or have multiple functions as scaffold proteins for RNA binding and regulation processes. And despite the minimization of the cells, the subset of RNA proteins or ribosome proteins has remained relatively constant. And we did this simply by cross-linking polypeptides, so proteins to RNA at using UV light. And with this, we've been able to generate a map of, or a list of 118 proteins that were found to be enriched when we compared them with a sample that was not UV cross-linked. And we can see how much they are cross-linked, which proteins are cross-linked. And what's important is that of the unknown proteins, 15 of them were found to have some sort of RNA interaction function. 439 and 505 are both in that list. And we then have three proteins, 440 added to these two, which are on the cell surface, which have two of which have RNA interacting abilities, and two of which fall within the acceptable range or recorded range of RNA nucleases or RNases that have been published for Mycoplasma species. So I was interested whether Andre and Chris had noticed RNA binding activity as well as the RNase or degradation activity that they saw in their experiments, because this could actually indicate that this set of three proteins form some sort of RNA degrading / transport mechanism for the incorporation of RNA nucleotides, basically, because there is no nucleotide synthesis biosynthesis abilities here in this cell, and the opt transport system is only for peptides. So it would be really interesting to flesh this out further, maybe with the CRISPR system, and we would be delighted to help out in this process. But this was a connection I could just make because of SynWiki and the data we have there and the two talks. So thank you for organizing this, John. Another project that we're working on is an inquiry into lipid metabolism here. In this species and in all mycoplasmas, there's only fatty acid acquisition and not synthesis, as we see in Bacillus subtilis. You have both. So the acquisition system follows the same as Staphylococcus aureus, where you have binding proteins B1 and B2 that have to be activated by kinases, and then they are taken up into the cell. But we have the fact B1, B2, and fact A protein in the Syn3 organism. But before fact A, we have an protein of unknown function, 0421. And
Yes.
we wanted.
Hi.
Okay.
Elizabeth, Via, can you mute yourself, please?
Okay, I think she's good. Okay, yeah. So we wanted to characterize a protein of unknown function that was before fact A, because we know from genome architecture in other organisms, that there could be a regulator before this process, because when lipids accumulate inside the cell, when they are taken up without regulation, this is toxic to the cell. So, and we checked via homology analysis, this 421 seems to appear to be an AS23, a member of the AS23 protein family, which are known to be involved in envelope stress envelope response regulation, this very broad umbrella term. And to begin characterizing this protein, we set about doing experiments like bacterial 2 hybrid experiments, which showed slight interactions between 421 and FAC-A, which we further confirmed using in vitro pull-down assays. And we could demonstrate that FAC-A and 0421 do in fact interact with each other in vitro. And we therefore think we have further solid ground, further evidence to go about exploring whether 421 can in fact regulate lipid acquisition in the synthetic organism, in Synth3A. And that's it for now. These are just two projects, but also the interaction projects are, so the mapping of the protein-protein interaction and protein-RNA interaction, these projects are still ongoing, and these are the two focus studies that I was doing. I'd like to thank my boss, Jörg Stulke, and the students that I've had in this process, the department here, which we got a lot of our techniques from, and the Impress for Genome Science, as well as our collaborators, Adam and Anna from Yuri's lab, and Alexander from the lab of Henning Urlaub here in Göttingen. If you have any questions, I'd be delighted to help.
What's the most interesting thing you've found so far, Neil?
Well, actually, it was the interaction that I just figured out today in your talk. That there's this new hypothesis for the three proteins, the 440, 439, and 505. And looking at what Andre and Chris just presented with their assays, I think this light bulb moment just happened, and I think that's really fascinating. And how it plugs in with SynWiki that Thiago put together, and the interaction maps that we're building with Adam and in Berlin, this collaboration really gave me a wonderful food for thought just now.
The data you showed will maybe help us narrow down our search for using the free gene set to see what might, I mean, it suggests that perhaps one of these proteins like 439 or, one of the proteins that you show may be the ribonuclease, the gene that has a ribonuclease activity that we have so far been unable to identify. And if it's a non-essential gene, this is something that we can drop from the genome. It would cost us some in growth rate, but still, this might be the key to enabling a cell-free expression system using mycoplasmas.
This could very well be the case. I looked it up actually while we were having the talks, and the 439 here is actually essential, and the other two are quasi-essential. And but 439, so 505 actually fits well within the range of nuclease. So they are supposed, for mycoplasma nucleases are supposed to be between 30 and 60 kilodaltons. So it fits within that range in terms of size. The other two are a bit on the high side. They are too large, so they could be binding or transport proteins. But all of them are lipoproteins, and all of them are uncharacterized. So these would definitely be candidates to start if you want to tone it down. And we were already thinking of approaching you for the CRISPR-I plasmid or the setup to try and work on the 421 things here. We could easily work on this as well and help out the community.
Well, send me a note and we will get the plasmids. I think Yo Suzuki is on the line, but we can get those plasmids in the mail to you. Those are easy to send since there's no, plasmids are easy to send.
Nice. This is awesome. Yeah, this is the most wonderful aspect of my work.
The comment, Neil, that was just brilliant. Thanks for pointing it out. We're just gonna be happy to check these proteins, these genes that you found. That will be That'll be awesome.
Yeah, cool. And if you need the plasmids, we can help provide 440 and 505 because these are not in those 398 genes, and it takes a lot of work or just time to get it done. Can save you some of that.
Okay, cool. Thanks. Let's keep in touch.